Excuse me, sir.
Yes.
Sir, can you please explain in short again that how the plaque assay works?
Uh, Black assay, I'm just, so virus infect a host cell, right? So as a result of that, if you have a solution where you are considering that you have virus particle, but you want to count the number of virus particle. Now that solution can be some sample like some sample from like patient or from even from I would say from water sample or any other thing, right? But it is taking time and it's like that's why like nowadays whatever that viral load you can see that is based on RT-PCR, it is fast, it is more time consuming. So here this sample you can dilute it like serial dilution, that part you understood. So now the host cell will be actually cultured, that means the host cell will be there on some petri dish, that means on some plate, right? And on that plate host cell will be in single layer. So if cells like sitting here, one cell, another cell, then you cannot see actually that plaque. So if it is single layer, that means just one cell here, another cell here. So as a result of that, a very low dilution of virus you put in this plate where you have this single layer cell, then at very like lower dilution, not higher dilution, that means very low number of virus particle there. Now say for example one cell here, one virus particle just infect this cell. So then what will happen, this virus will increase in number inside this cell, and then whenever after the eclipse period, that means when the viral particle will be synthesized inside the cell, the viral genome or capsid, then it will rupture this cell and it will come out and it will infect other cells present on that plate. But we don't want that. We want to restrict that. That's why on top of you have single layer of cell, you added viral sample, right, for infection. Then you put some semi-solid layer so that when the eclipse period will be over, those virus particle will not travel on the plate and it cannot infect other cell which is present in a long distance, like in a different portion of the plate. Only it can infect just adjacent cell. So as a result of that, what will happen? That if you have one virus here, it will actually destroy this cell and the surrounding one or two cells or four cells, something like that. So as a result of that, in plate, in the petri plate, you have like monolayer cells, but you have many cells in the petri plate. Maybe 4 or 5% you actually found that those portions, in those portions cells were destroyed. Which cells? Host cells. How? Because of this viral infection. How you will see that? You cannot see virus particle through open eye or through through light microscope. So we have some technique. We add some solution where live cell, that means our host cell, cells which are live. So they will be like they will take some dark blue stain. So as a result of that, in some spot, you will not get the blue stain. Why? Because those cells are already ruptured by viruses. So as a result of that, you will get some kind of spot, like something like this here. So this is real experiment, some spot like this. This means you have one virus particle here and it causes this plaque. Another virus particle here is causes this spot. That way you can understand that whatever sample you added there and how many virus particle was there. So as a result of that, you can actually find out in your stock solution how much virus particle there. But the question is why we are doing this serial dilution? Because if you take from the stock solution, the number is too high. So all cell present in monolayer, that means all host cells, everyone will be, all will be infected by a virus particle. So everything will be plaque. That means no live cell. So you cannot count, right? So as a result of that, you cannot tell the number. So as a result of that, you have to take very like little number of viruses. That's why we are doing serial dilution. And whatever the dilution factor you use to do this experiment, then later on multiply and calculate what is the the number of virus particle present in the stock solution. Rajesh, is it clear?
Yes, sir. So we observe one cycle of growth of viruses, and for each dilution step, and then we generate a formula out of it and predict the number of viruses, kind of like
Yeah,
that.
so the serial dilution is nothing, just few minutes. Just virus sample, you are diluting. And now from with each dilution, like whatever each dilution you have, you can use all of them in a defined experiment. But the problem will be in very, like I would say, where virus concentration is too high, you cannot count anything. Only in certain condition, like where you don't know the initial number of viruses. That's why we are doing this serial dilution. In certain dilution, you will see proper number of plate, what you can actually calculate, right? So that way you have to calculate. So that's the very traditional technique how we count the number of virus particle. If you have additional question, Rajesh, maybe this is very standard thing actually. Even you can, I believe, you can search in Wikipedia or somewhere that viral plaque assay and go through it. Then, if problem, let me know.
Yes, sir.
Yeah. So now I will go into a different, portion like, this is done about, I'm done about, virus growth, plaque assay, and little bit of viral life cycle, whatever I mentioned. Now, like two new terms, equilibrium and non-equilibrium viruses. This is interesting. If you see, equilibrium viruses have been associated with the host system for a very long period of time. They are usually non-lethal but spreads well. Example, common cold. Equilibrium virus means they are staying with the host for a long time. So that's why they are not lethal. That means if the host, see, traditionally, the idea is if some viruses are very lethal, they shouldn't spread oil because that means virus is infecting some individual. Forget about human. Like virus can infect many other animal, many other plant also. If they are too lethal, so whoever it infect, that particular organism might die. So as a result of that, transmission should not be very high, right? Similarly, some viruses which are not very lethal, say for example, common cold, rhinovirus, common cough, cold, sneezing, that is very common. All of us we suffer like 2-3 times in a year, like common cold. So that virus is equilibrium virus. They don't like cause some kind of big problem to host. So that is good for them also, so that they can be with the host for long time, no problem, right? Because they can transmit very quickly, but they are not lethal. They will not cause like a lot of problem to the host. So this is called equilibrium virus. And as I told you, like common cold, rhinovirus, and non-equilibrium virus have recently jumped from another species. So as I told you, virus are species specific. That means some virus, they are only infecting human. Some virus, they are only infecting some bird. Some virus, they infecting some fish, right? But non-equilibrium virus means they have recently jumped from another species, but they are not associated for a long time with human or any other specific host. They are sometimes lethal, may spread poorly or well, and represent most of the deadly viruses. Say, for example, SARS-CoV-2. So what do you say? This is equilibrium or non-equilibrium?
So
So they
non-equilibrium.
spread, good, however, even if they are non-equilibrium.
Yeah, this is non-equilibrium because this is lethal actually, because common cold is not lethal actually, because whether the percentage is low or high, but this is lethal. So this should be categorized under non-equilibrium viruses. Okay. So, and also this is new also, like they are not staying for long time. This is recent origin, whether it's from another species or somewhere else, but it's a recent origin. Hmm.
So, but
Hmm.
Sir,
SARS-CoV-2 spreads very quickly, so that characteristic of non-equilibrium viruses will
Yeah. Yeah, so non-equilibrium virus is actually like that's why I mentioned there in the slide that the spread like equilibrium viruses they spread fast, but non-equilibrium viruses they can have the both property. Either they can fast very fast or they can spread slowly also. Right. So for example, in this case, yes, it is spreading fast. So now, if you see here, the equilibrium viruses, for example, polio. Polio was there, but only few percentage of people they have little bit of problem in nervous system with polio. But apart from that, if you see common cold like coronavirus, adenovirus, as I told you, that old coronavirus, that means traditional coronavirus which cause common flu, nothing wrong with that. And measles, herpes, those are equilibrium viruses. On the other hand, HIV, Ebola is very dangerous virus. Influenza, SARS-CoV, those are actually non-equilibrium viruses. I put this back because still it is in debate whether because coronavirus like they are like bats, they are reservoir of coronavirus. Lot of different types of coronavirus present in bat. Okay, so that's why I just put this, but still there that debate. And there are,
sir, can you go to the last slide once?
Sir,
yeah,
sir, coronavirus is a type of common cold.
coronavirus. Yeah, so 1 minute. There are two questions. One is last slide, and I went there. And yes, so as I told you, the coronavirus, I actually showed you that electron micrograph image, like at the beginning of this class. That is long time old, like
Sir,
you
excuse me,
like.
sir. Sir, if you don't show it, then there's no point because I can't see it. You
No, no,
switched it and just change the
no,
thing.
no. I'll show the two questions there actually. So I'm addressing that thing also. I don't know whether the same person you asked the same question or not.
Sir, I said about the PDF. If you can show it once. Thanks.
So you are saying asking about this.
Yes, sir. Thanks. One second.
Yeah. Tell me what is your question.
Sir, I was just noting it down. I didn't have any question.
Oh, okay. But I will send slide, no problem. So everything you will get right in your like MS Team, like even like same day I upload. Okay, so after few hours you will get it. Okay, no problem. So, and another student asked about coronavirus, whether this is common cold. Yes. So, like 1 minute, because I have many things to discuss actually. Ronit Datta. So please, you don't need to write actually. Okay. So I will send you slide. Just after the class, within few hours you will get it in your MS Team. So no problem. And so here, yes, coronavirus is also causing common cold, and that's why you will see that I'm going a little bit in a different direction for this discussion. This is required actually. Many people there, although they are even infected with this, SARS-CoV-2, but some of them are nothing like nothing happening there because most likely they are actually infected before by similar kind of coronavirus, not by SARS-CoV-2. That's why their immune system already aware actually about this, some of the information because they are linked, they are similar. This logic you will get more in my next class in the immunology class. Okay, so there are a number of human coronaviruses which causes common cold with mild flu-like symptoms. Ideal example of equilibrium virus. But this SARS-CoV-2 is not an equilibrium virus. That is causing the problem, right? This novel coronavirus. So now, non-equilibrium viruses, as you can see, that natural host, wild aquatic animal, mostly bird in case of influenza, and HIV. All of you are aware about HIV. Their natural host is chimpanzee, and coronavirus, Nipah, Ebola in bats. So Nipah, I believe that few years back, some Nipah cases were reported in Kerala, and it was a deadly virus. but it's immediately stop also. Like, and that's all. So those are non-equilibrium, example of non-equilibrium viruses. Just get the idea about non-equilibrium and equilibrium viruses. That's all. So,
Sir, I have a question. Sir, why the term equilibrium and non-equilibrium? I couldn't understand the
Yes, sir.
terminology.
Same question. Same question. I also have.
yeah. So I believe this is not you shouldn't make it this equilibrium non-equilibrium with your physical or chemical reaction. So equilibrium viruses here they are stating that equilibrium with host. That means they are causing infection. Little bit of, as I told, mild cold. What is happening? You are going to your school, going to your college, you are doing everything. Virus is also happy, you are also happy. Nothing, no problem there itself, right? Non-equilibrium viruses, they are not in equilibrium. Either virus should not be there, like you have to get cured by either by medicine or whatever the way, your immune system, whatever the way, or the host is, suffering. So it's a lot of suffering. So non-equilibrium, they are in equilibrium with a host actually. They are always there from very beginning. I would say like hundreds of years they are there, but they are in equilibrium. Is it clear?
Yes, sir.
Yeah. So now, if you see coronavirus infection, human coronavirus were first isolated in 1967, causes common cold with mild symptoms like sore throat, fever, headache. Coronaviruses from other animals have recently started crossing the species, and that causes, infection. For example, a recent coronavirus outbreak. If you see, this is mostly I have taken from Wikipedia or online sources, not from textbook. So SARS coronavirus. I believe all of you know about what is the full form of SARS. Severe Acute Respiratory Syndrome. SARS, Severe Acute Respiratory Syndrome. Coronavirus epidemic in 2002 and 3. Only few cases and number of death was reported, whatever I found in website. And then MERS, this is Middle East Respiratory Syndrome. Coronavirus epidemic in 2012 and 13, and few cases and few deaths were there. And now novel coronavirus here, this is the new one, a lot of cases and a lot of death. But if you see the fatality rate, here more actually in case of SARS-CoV, SARS coronavirus in 2002 or MERS-CoV compared to here, the fatality rate is much less. But here, like although I am showing this slide, I would like to discuss with you. This fatality rate percentage, to me, it doesn't make that much of sense because, none of those by corona, like, MERS-CoV or other, like whatever I mentioned, that time, like nothing like this kind of test was going on. People are, this is sometimes I'm like, puzzled that what is right or what is wrong. Because now every day, even in India, approximate test is around, say, 17 lakhs test for coronavirus, right? So immediately you are getting more and more person, they don't have symptoms or they are not suffering, but you are getting positive result. So as a result of that, the number of fatality days also decreasing. So a lot of, I think all of you are enough intelligent to understand this. A lot of, whatever we are discussing today, like today means not in this class, I'm saying that discussing in today's pandemic situation, many of those things are little bit, in terms of data, which one you have to, believe or which one you should ignore. There are a lot of issues there, right? So as a result of that, here you can see only fatality is 2.2%, but that might be because of, we are taking a lot of tests, right? And if you do the same kind of test here also, you might get the similar scenario. Who knows, right? So now coronavirus, as I told you, a non-equilibrium virus. So in case of MERS-CoV, Middle East Respiratory syndrome. So you can understand Middle East. So the animal responsible to bring it human is camel, and in SARS-CoV 2002-3 most likely it was civet cat. So from bat to this civet cat and finally to human, and this one we just put some sent like here some question mark because of whether it came through directly to bat or through some lab. There are a lot of questions there. Okay, so I'm not going into that debate now. Let us discuss.
That
Yeah,
it was due to some pangolin or some kind of animal like that.
pangolin, right?
Yes, sir.
Yeah, that's what I'm saying. There are a lot of issues there. So in recent times, I'd like to mention, if you are like little bit getting more interest after this class, little bit study about the origin of this, this SARS-CoV-2. Also, you will find that many theories, and some of them based on experiment. Also, not like not like theory just by thinking. Some based on experiment. Experiment means based on the genome sequence. That some genome sequence, say for example, I'm telling you one thing. Just think about it. So if you like you already attend genetic engineering classes, right? If someone do lot of tinkering, then if you sequence the genome, some unnatural thing you will find. Like coronavirus, they should have some kind of similarity with other coronavirus. But if you get some kind of manipulation, I'm just giving you example. So for example, some unusual site, some kind of restriction site, that means some people actually tinker with that. So but this is not proven. Actually proven means some experiment shows that, and as I told you, this is some debate, and you guys are listening a lot of news, so we will we have to see. So now here the severe acute respiratory syndrome or SARS-CoV-2. So for example, in this case, you can see this is how SARS-CoV-2 looks like. Okay, and this is spike protein, and inside the virus they have the genomic RNA. Apart from spike protein, some other proteins are also there. And finally, if you see how it infects host cell, so this is electron micrograph, right? So this is nothing but you cannot see anything more than that, and this is just some reconstituted image. So if you see here, how does SARS-CoV-2 infect human host? So this is SARS-CoV-2. This is the viral particle here. So this is the virus, and now this is spike protein. Spike protein, and if you see in the spike protein, there are many regions. I'm not going into the details. It's not required to know. But the idea is what happened. I believe most of you know, almost all of you know what is the current or known receptor for this virus. That is called ACE2. Even in newspaper you will get it. ACE2 of human host. ACE2 is another protein present on host cell that is the receptor for this virus. What happened? This spike protein interact or binds to ACE2 receptor. As a result of that, this interaction leads to endocytosis of this virus. That means that virus go inside the host cell. So here I have taken from some review article. This is recent, you understand, right? This year most likely. So now you can see that this is actually cell membrane. Whose cell membrane? Our cell membrane, host cell membrane, okay? Cell membrane. On the cell membrane, you have this protein that is called ACE2. Now, this virus, they have spike protein. We are just zooming, like, we are showing in large, like one spike protein here. And now this spike protein, they specifically interact with this ACE2. This is receptor binding. That means receptor and the viral protein, spike protein, they bind together. And finally, that leads to endocytosis. Here you can see that leads to viral entry inside the cell. So this blue color part is the inside the cell, host cell. So now the virus is inside the cell, and finally this is your viral genome, and here that complexity is there, that this is single stranded RNA. So which strand is there, based on that another strand is being synthesized. Please don't go into that details. Within this short period of time, it is like almost impossible for me to explain all those things. But from this RNA, you will get, this is, in this case, no reverse transcriptase is used. This is, see, the enzyme is here mentioned, RDRP. What is RDRP? RNA dependent RNA polymerase. Again, I am saying RNA dependent RNA polymerase. This is the enzyme that synthesizes the RNA of SARS-CoV-2. But how this RDRP you will get? In order to even get it, you need to translate some of these, RNA, which is coming inside the cell. And finally, I'm not again going into the details of all these steps, but finally you will get all these proteins required to assemble the virus again. That means spike protein itself, as well as other protein, whatever present. Apart from spike protein, some other protein are also there, and as well as genomic material. And finally, they will assemble and they will again come out from the cell, and those virus particles, they will infect nearby cell. They will infect again other cell. So that way, SARS-CoV-2 infect cells. But this is true for other viruses also. The only thing, these portions become different to virus depending on their genomic material, whether they have RNA genome or DNA genome, or which kind of strand of RNA, whether this is sense strand or antisense strand, based on that. Okay.
Sir, how
Sir,
does it
why
get
are
through the
the
cell membrane?
Sorry, say again.
Sir, how does the virus get through the cell membrane after binding the receptor?
Yeah, good question. Good question. So, because I believe that you are asking because they don't have envelope. I told that envelope they get fused. So, see, again, I would like to say almost all viruses, like for example, measles virus and so many viruses, their policy is same. They actually, like, that's why if you say that what is the function of ACE2 receptor? ACE2 has different function, physiological function in our body. It plays some other role, physiological role that is required for us. But most of the viruses, they hijack our system. Hijack means, they are utilizing the function of ACE2 cannot be that they are recruiting virus particle. That cannot be function. So similarly, like measles virus, HSV virus, lot of other viruses are there. They use or they hijack some of our receptors, some of the cell surface receptor present on our cell surface, right? They cleverly hijack, they just bind it. And I'm not going into the details of cell biology. So in this region, I actually work in my own interest there also, that when something like outside particle, something that bind to receptor, and that interaction leads to some kind of endocytosis. Endo means going inside, endo. Cytosis means carried out by, cytosis. They are going, like, in school days, you actually studied about phagocytosis, pinocytosis, that means cell eating something. So similar term, endocytosis, it is going inside the cell. That means actually here the mechanism is host cell itself engulf this thing. Some, like some cytoskeletal material present in host cell, they get remodeled. They actually, I'm just showing you here. So what happened here? You can see here, budding here, the similar thing. So for example, whenever this interaction happen, then the cell membrane, they will be like this, and finally they will be like this. That way they will engulf the virus, and virus will go inside the cell. And then the viral genome will come outside, and it will start making more and more particle. But whoever asked this question, I didn't see that time. But the major idea is this endocytosis is very common. This is true for even some bacteria, like some intercellular bacteria, when they infect, when they bind to our host cell, and that help to endocytose that bacteria into the host cell. That is one of my research project. Even if you go my website, you will see that. So again, I told you before that I am not what I have no experience with working with virus, but one of my project deal with the endocytosis of bacteria inside the host cell. Okay. So, but the overall rationale is same. So now,
Sir, can you go to the slide once?
yeah,
Sir.
yeah,
Sir, here, here it is given that like after the viral entry, there is one is replication and packaging, and another is transcription. But like both pathways are present. I don't understand why transcription is there, because it is an RNA virus. So
yeah, yeah. But yeah, it is required because see, a very good question actually. But in short time, I didn't explain all those things. But see, whoever asked this question, very good, because that you are saying that replication should be enough. We can do replication first, right? But the thing is, in order to replicate, you need this RNA-dependent RNA polymerase. This is not present at the moment in this virus. They have only spike protein, some other protein present in the capsid, right? But they have the information in their genome to make this RNA-dependent RNA polymerase. Then you have to make that protein in order to even replicate its RNA. So as a result of that, they are transcribing their RNA, whatever present here, they are getting mRNA. This mRNA will be translated by host machinery, and then after translation, you will see that these proteins are coming, and those proteins actually now help to synthesize its own genome. Is it clear?
So the transcription occurs on DNA, right? So how can it occur on RNA?
Okay, I understand your question.
yes, sir,
Yeah.
this was my question.
Yeah. So yeah, okay. I got your point. So this is actually I will see this review article. Actually, your question is absolutely fine that we shouldn't like write here the transcription here. That is absolutely fine because this is I have taken it directly from one your review article. But your question is very much valid. Whatever until now I taught, we shouldn't say this is transcription. This is true. Okay, so then ignore this transcription step. So only thing this conversion between this mRNA, like this positive sense to negative sense, that way conversion is going on. But you shouldn't say that as as transcription. Whatever we actually learned before, you are right. Actually, we should ignore that. I will check that, and then I will post this thing in MS Teams. And even I can post this review article also. Like I have already downloaded. I can post that also. If someone of you are interested, you can just go through those figure and figure legend also, right?
Yes,
And
yes.
yeah. So now HIV virus, I will take 15-20 minutes, not that much. So we are not this time not like discussing in details about HIV. I will like tell a little bit about that. Mostly we spend a lot of time about this SARS-CoV and other thing. So HIV is a classical non-equilibrium viruses. The problem here is, see, HIV is a human immunodeficiency virus, and the disease is called AIDS, acquired immunodeficiency syndrome. The idea is this virus, what happened, what are the host cells, like HIV, they infect human now, right? But what are the actual cell they infect and they destroy or they down-regulate their function? Are we aware?
Sir, WBCs.
Yeah, WBC, right, very good. So in your level, it is good WBC you told. It is kind of CD4 cell. CD4 is a certain population of immune cell. Actually WBC cell, you are right. Red blood cell, WBC cell. Some of those WBC cell are actually our immune cells, right? Actually, this virus, they actually target those immune cell itself. And this CD4 cell, in the next class, I will discuss in more details. Those are huge, I would say, very vital immune cells in our body that protect us. If now those viruses, they are creating problem in those immune cells, then what will be the effect? Our immune system get dampened. Our immune system get very, very weak, right? So that is the reason that disease is called acquired immunodeficiency syndrome. That means because of the viral infection, someone acquired what? Immune deficiency. What the deficiency? Immune, right? The immune system is low now. It's not functioning properly. And that is the reason like HIV like infected people or AIDS, like people with AIDS, they normally get died with some other disease because their immune system is very much downward now. So as a result of that, that person might get infected with many other diseases because of many other infection, and that causes more problem. So here some life cycle there, ignore this slide. Okay, I will delete that and then send it to you. So you don't need to go through it. So here kind of then you will be kind of totally puzzled also. This is I just mentioned here. Please don't go much more details. If you are only some of you interested, then only go. Here, like some viruses are there, particularly this HIV virus, even they can integrate their genome to our host genome. Again, I'm telling you, I'm not going into the details, but some viruses, they can integrate their genome into host genome. That's why that HIV, if someone get HIV, that information, that genome is actually integrated with host. So that's why HIV is there. Only the remedy is the
That
lifelong.
means HIV is a DNA, like it contains DNA as its genetic material.
See, that depends. This is normally, there are like big classification that is called Baltimore classification, that David Baltimore actually, Baltimore classification. So he categorized this genome into 6 different. It's better like we shouldn't discuss that much of details, but most of the DNA virus, they don't integrate their genome into our host. It is actually RNA genome. But still, that's why I told you that we will not discuss this. Okay, I will delete this thing. But if you are interested, write me in my institute email ID. I will send you proper like review article or something like that. Or we can you can write me in the like chat section of this MS Team, and I will frequently check the message. I will answer, and we can continue some discussion if you are interested. That means this is for normal interest, not for exam or anything. But please write me in the chat box. Okay.
Okay, sir.
Yeah. So I will delete those slides
Excuse me, sir.
because yeah.
Just a small doubt that non-equilibrium virus, do they means directly kill the host, or it also includes the viruses that indirectly kill the host? Because HIV does not like directly kill the host; it weakens our immune system.
Yeah. That your question, I get it. So as a result of that, Rajesh, so I think in every case, similarly, like someone say, like one person can say, even for SARS-CoV-2, actually virus is killing us. But the thing is, finally, that disastrous thing is happening because of cytokine storm, our own immune system, right? But how you can do? You cannot reduce your immune system. Then you will, someone will get some other infection also. But in this case, particularly in case of HIV, the thing is we didn't discuss in much details. Actually, that is one viral life cycle. That phase is called latent phase. That means virus is always there, like that genome is already integrated. But the problem is slowly, like over the time, like all the time, they are actually infecting some of the population of our white blood cell, as Soham actually told that, particularly CD4 cell. So our immune system is weakened. So as a result of that, we get secondary infection, other diseases also. Mostly the combination between HIV and TB is dangerous. This is the major cause, like HIV and TB together, it's kind of really dangerous thing because TB is also very much problematic to get rid of. Most of us, many of us, we have TB, right? But the thing is that is in latent condition. We are healthy enough. Our immune system is healthy enough so that we can like that is not showing any symptoms. Again, listen that this is very important message that many of us, we have actually some kind of mycobacterial infection, latent infection, but we don't see any symptoms. We are still good. We are eating properly. We are, continuing good, habit like, healthy life, no problem. But if someone suffer from some other issues, some like, immune system is low during that time, that's why this TB, they are called sometime, that if they get the proper opportunity, then they will, causes more problem. I think many of you know, right? In particularly in tropical country in India, almost 1/3 of our population we have actually TB with us. But we don't
Yes.
show symptoms. Yeah, we don't show
Yes.
symptoms. Yeah, but these two combination will be deadly actually. So anyway, this is almost done. Just one slide. I will briefly discuss about that.
Sir,
What is yeah.
why are we not able to create a vaccine for HIV?
Very good question actually. Why that is true for almost all RNA virus. So as I'm saying, almost all RNA virus. So RNA the vaccine against RNA virus is not good. Now you will ask me that, sir, why then we are like talking too much about this Covishield, Covaxin, and so many things. Actually, if you see